How might scientists determine which genes' mutations are responsible for a given cancer? Well, one could compare gene expression between normal cells and then cancer cells from that tissue type. So, for example, compare normal breast cells to those of breast cancer cells. In this first example, where the insulin receptor is being studied, both the cancer cell and the normal cell have about the same expression pattern, and so this gene does not seem to have a role in the cancer. Aromatase, the enzyme which synthesizes estrogen, is in this case at a higher expression rate in the cancer cell, suggesting that it promotes the abnormal growth of the cancer. In these two examples, cyclin D is being expressed in the cancer cell but not the normal cell. And the anti-apoptotic BCL2, a protein which can prevent programmed cell death, is being expressed in the cancer cell but not in the normal cell. And so these proteins may have roles in promoting abnormal cell growth. In this cell, the normal cell is expressing a tumor suppressor BRCA1 protein that the cancer cell is not. So it may be that this protein's normal role is to prevent cancer, and that the cancer cell has inactivated it. In the same way, this normal cell is expressing more TGF than the cancer cell. This TGF receptor protein may have a role in limiting cell growth. And finally, while both the normal cells and cancer cells are expressing the estrogen receptor and p53, there are differences. And so therefore, the mutant protein may not function in the same way that the normal protein does. And so in this case, one of the proteins is localizing to the nucleus while the other one is not. So it may be that the mutation changes the function of the mutant protein. Here it can be noted that different cancers can express different mutated genes. Just because the mutated genes in cancer 1 from patient 1 are these, a different set of mutated genes may be expressed in the cancer from patient 2. And cancers often express multiple abnormal genes. Very often one mutated gene alone is not sufficient. So it would not be surprising if there are multiple genes whose expression patterns vary from normal cells. And finally, there may be differences in the expression patterns in cancer cells from early in tumor progression to those later in tumor progression, which may mean that the treatments which might be effective early in a tumor's progression might not be as effective later.